All right, good morning. Starting Unit 6 today, we take a look at the inside of cells. So we were looking at the outside of cells, the cell surface, in our last chapter, chapter 5. And so now, Unit 6, we move into the cells and we look at cell organelles. All right, so first we'll note here, we've got two types of cells. Prokaryotes are cells that lack a nucleus and other membrane-bound organelles. So I like to specify membrane-bound organelles. Kind of depends on how you define an organelle. For example, ribosomes, prokaryotes have. So if you call ribosomes an organelle, well, ribosomes have some organelles. Flagella, the tail-like structure. I don't know if you guys remember, we covered a lot of these additional structures back in chapter 1, I believe it was. So make sure you're going back there. I know your first homework kind of works with that. Kind of go back, review some of those basic structures. You're going to see them again on this chapter test because they are organelles and they fit in here as well. And hey, no better time to review for a test than we got a final coming up, so good time to start throwing a review out there. All right, the other type of cell, and these are more like our cells, these are eukaryotes. So eukaryotes are so named because they have a true nucleus. All right. How did we get eukaryotes from prokaryotes? That is answered by the endosymbiotic theory. Here's the idea of where eukaryotes or how eukaryotes emerged from prokaryotes. So one bacteria remember prokaryotes are bacteria are prokaryotes. So one bacteria ate another one and they work together. And that's the theory. So endo, kind of like endocytosis, we covered that last chapter. Endocytosis, one bacteria kind of endocytosed another one, and then they work together. Symbiosis, two organisms working together. And so that's our theory for where cells that have organelles came from. So there's one cell. There's a different color here. And draw a second cell. Again, like we just kind of mentioned, it's a lot like endocytosis. We have the one cell kind of swallowing the other one. And that's how we got organelles. And there's three key pieces of evidence that kind of go along with this. Number one, a lot of our organelles, and we usually talk about the mitochondria and the chloroplast, but a lot of organelles are the same size as bacteria. So our cells, eukaryotic cells, the cell that ate the other one, they've grown, they've gotten larger, they've gotten more specific with what they do, so they've gotten bigger. But the organelles inside of them are still about the same size as a bacteria cell. Two, this next one I like a lot. Like this really cool. Mitochondria and chloroplasts have their own DNA. So they're separate from the nucleus, actually make their own proteins. And to boot, it's bacteria-esque DNA too. It's a lot like bacteria you would find in a bacteria. So mitochondria and chloroplasts have their own DNA. Well, that's interesting. And finally, then, third piece of evidence, many organelles have two membranes. So looks like a duck, quacks like a duck. Oh my goodness, have their own DNA. And this kind of explains this right here. I mean, how many membranes would you expect an organelle to have if it were indeed swallowed by another one? Have two membranes, right? Because the outside of the blue cell and well, the inside would be the blue cell, and then the outside is the cell that was swallowed. So, man, it kind of helps to explain why organelles have a lot of times two membranes. Pretty cool stuff. All right. All right, moving on to the nucleus. We're going to spend some time taking a look at different organelles. We'll start probably the most recognizable one, nucleus. All right, so roughly the shape of a circle. Now you got to think it's more like a sphere. It goes all the way around. So we've cut it in half, and we're kind of looking down into it. Actually, hopefully we're not writing in pen. I'm going to erase this section here. We're going to add on to this right here in a second. Right now I'm going to blow this area up, so I'm going to make it a little bit bigger here so we can actually take a closer look. All right, so this here is what we call the nuclear envelope. and it is made up of a double phospholipid bilayer. There's one phospholipid bilayer. Here's the second. There's that double membrane we were just talking about on that last page. We've got kind of a double membrane here. This right here is a nuclear pore. So they're integral proteins. We learned about these guys last chapter. Proteins that span the entire, in fact, they span not just the entire membrane, they span the entire double membrane. And they allow things into or out of the nucleus. So very important, we have our nuclear pores to let things into and out of the nucleus. Now it's going to be really tough to get into or out of the nucleus. That's like the highest secure area in the cell. So not a whole lot of things can get into or out of the nucleus. We'll take a closer look. This is what we call the nuclear envelope. Again, it's a double membrane that surrounds the nucleus. All right, this here is called the nucleolus. I'm actually not sure why we use the same word to so many different times in the same setting. Nucleolus is different than the nucleus. So we're looking the whole thing together. All these pieces are adding up to the nucleus. But this right here is the nucleolus, which is found inside the nucleus. And I'm actually sure why it's found in the nucleus. It's a collection of proteins that their job is to make ribosomes. Now, ribosomes, we know, read the mRNA, the copy of the DNA, and translate it into protein. But that doesn't generally happen inside the nucleus. So I don't know why we make the ribosomes in the nucleus when we don't use them except outside the nucleus. So it's kind of confusing, but yeah, something more to remember. The nucleolus is found inside the nucleus, and that's what makes or where ribosomes are made. All right, next up, what looks like a big plate full of spaghetti. Chromatin. This is the genetic material. It's made up of DNA and proteins that help to organize it. And it does look like a big plate full of spaghetti generally. We'll talk later about when it condenses and it becomes a form that we can actually see. But yeah, for now, it really, chromatin just looks like a big plate full of spaghetti. You couldn't tell where one strand ends and another strand begins. And finally, one last thing to add, and that's why I left this part over here kind of open. Not on the screen anymore. There we go. So this is the endoplasmic reticulum. And there's two types of endoplasmic reticulum, the rough ER is the site of protein synthesis. And the smooth ER makes lipids. So there's two types. We'll talk more about those two types a little bit later. Just wanted to show the endoplasmic reticulum, not really a part of the nucleus, but it is the membrane of the endoplasmic reticulum is continuous with the nucleus. So that's why I wanted to show that here. In fact, to be most accurate, I should probably draw another one up here doing the same thing and another one down here. The endoplasmic reticulum really completely surrounds the nucleus. It's all over the nucleus. Again, two types. The rough ER is the site of the protein synthesis. Again, we'll talk more about that later. And the smooth ER is where lipids are made. One thing I forgot to do, and I go back to the very beginning of today's notes, give this section a title, give the unit a title. How about the section? This is 6.1. And we covered here the endosymbiotic theory and the nucleus. All right, we'll be back tomorrow for 6.2. I'll see you then.